Okay, good morning, everybody, and welcome to the All Ireland Cancer Research Institute's Immuno-Oncology and the Microbiome workshop. We're very pleased to bring everybody together today for this really important event to bring together researchers across the island of Ireland who are interested in immuno-oncology and the microbiome to come together to share expertise and potentially to create thematic goals as a group with which we can support translational research as an island into the future. To introduce this concept and where this pillar might fit in, I'm very pleased to welcome Liam Gallagher, who's spearheading this group, and he's going to show us some slides and a video, and I'll hand over to Liam.
Yeah, hello everybody. Thanks very much, Jeruska, and I'd like to also acknowledge Paul O'Toole and Subrata Ghosh from UCC as well, who are the kind of main organisers, along with Jeruska Nadu from RCSI, and I'd like to also thank, in particular, Isabella in the background in terms of logistics, and also Rosemary. So look, I'll just show a few slides. Just kind of, I suppose, some people might have seen, understand the context between this concept of the All Ireland Cancer Research Institute. But for those who are not familiar, I have a couple of kind of introductory slides. So, Jeruska, can you see those? Okay. Okay, that's great. Okay, again, I suppose what I'm trying to do here is just give you a kind of concept overview of what we're trying to achieve, which is really fundamentally bringing people together to focus on cancer research on All Ireland, to really develop an overarching framework for cancer research across the two jurisdictions, but both north-south, but also east-west. So again, it's interinstitutional collaboration on the island. So when we talk about AICRI, or the All Ireland Cancer Research Institute, really what are the kind of nuts and bolts kind of concepts here? Where we're talking about a cross-border program or cross-institutional program connecting cancer researchers of all different types across the island. And really what we're endeavouring to do is reduce fragmentation in terms of research, diagnosis and treatment. We're not trying to replace or like obviously each institution is probably developing their own kind of framework in terms of cancer research or interactions with, between academic institutions and their affiliated hospitals. What we're trying to do is provide a connecting gel between those different institutions. We're quite keen on building critical mass in specific areas, particularly in the area of precision medicine, personalised health. It's not a bricks and mortar program, at least the concept behind AICRI. It's really a virtual institute with linked facilities in terms of scientific facilities, both north and south. A lot of the focus is really on kind of, I suppose, taking advantage of, the power of data. We also think by creating the kind of AICRI concept or the initiative that we can create a gravitational hub to attract people into research in the country. And we think there's some specific opportunities in particular areas, for example, looking at the interconnection between cancer and comorbidities. So just it's a short video, which is a kind of which we developed about a year ago, really to summarize for multiple stakeholders what we're trying to do within AICRI. So I'm just going to play this now.
On the island of Ireland, 1 in 2 people on average will develop cancer during their lifetime. People's lives change overnight. A cancer diagnosis brings huge worry and anxiety not only to patients, but to their families also. We want to help patients live longer and better lives after their diagnosis. We will connect cancer researchers across the island of Ireland. We will work alongside patients to make sure that their voice is heard. By coming together, we can harness all of our different skills and expertise to better understand cancer, to develop more personalised treatment options, to ease suffering and save lives. This is our vision for the All Ireland Cancer Research Institute. We will work hard to uncover better diagnostics and treatments for patients. We will learn more about preventing cancer as well as tackling key issues related to living with and beyond it. We will become a global leader in cancer research, and as a result, we will give much needed hope to patients and their families. Please join us in our mission.
So thanks to Aidan Gillen who did a voiceover for that, and really that's I suppose a rallying call for the overall vision behind the institute, which we developed, I suppose, over the last year and a half. And in broad brush strokes, I suppose what we're trying to do is, I suppose, change the scene in terms of how cancer research is supported on Ireland. Classically, a lot of cancer research programs were funded on, I'd say, a 3 to 5 year kind of cycle. What we're quite keen is to actually consider a kind of longer time frame in terms of taking on board, cancer is a kind of long-term kind of investment in terms of research. And so we're looking at a large-scale program in excess of, say, 50 million euro, which have three kind of broad thematic areas. One, data-driven discovery science, bottom-up discovery science. We've received some funding to actually support that area just recently, which I'll cover in a few minutes. We're also quite keen on onboarding innovations, particularly into the healthcare system, or improving that kind of cycle of innovation or discovery and then embedding within a clinical environment, particularly in the area of phase 1 clinical trials, and I suppose looking at the potential power of AI approaches. There's also kind of, I suppose, some good work which has taken place in terms of biobanking within Ireland, but really needs to kind of, I suppose, be further resourced. And the other component is really, I suppose, connecting and creating an ecosystem for innovation. So we have a burgeoning industry ecosystem, or kind of community of companies, both small and large, with interest in oncology in Ireland, either diagnostics, therapeutics, or allied areas, and we're kind of keen to kind of nourish that going forward. So I suppose what we're doing is creating this overarching framework, and this research workshop is one kind of key component of that. Is to kind of we define this as a kind of key area of opportunity. And what we're quite keen on is we'll go into the community, understanding what the current state is in that area, what are the opportunities, and how do we push that forward. So I suppose what we've done over the last year and a half is we've engaged, particularly, and we're still in this engagement process within the academic environment, really look at kind of the key researchers which are out in the space, key topics, what are the infrastructural requirements. We've also engaged quite closely with governmental agencies to kind of, I suppose, advocate for the AICRI concept, and we've gotten good uptake in terms of kind of interest in that area. We have next week a patient stakeholder workshop taking place as an aside to the AICRI meeting, which I'll discuss in a minute. And also we're planning an industry workshop later in the year. So for AICRI, we need all of these stakeholders involved in kind of defining the agenda. So approximately about a year ago, I suppose what we set out is to really, again, lots of good cancer research going on across the island in different institutions, and really we want to come up with a kind of an interconnecting collection of institutions. So we have 10 institutions, academic institutions on board, 2 in Northern Ireland, 8 in Republic of Ireland. And also we have other stakeholders. So we have kind of key representatives from those institutions involved. And then we have, I suppose, participation from other kind of key stakeholders. And this will grow and evolve over time in terms of as we mature the concept. Just some highlights in terms of, I suppose, what we've done over the last year. We had an early initial success, a modest amount of funding, but it was quite critical funding, I suppose, to kind of look at defining our kind of areas of interest under the IRC New Foundation Scheme. This is part of the Shared Island Programme. And this is covering a series of workshops, which I'll cover in a minute. We also had success under the SFI Research Infrastructure Programme. This is just one example of a national spatial tissue profiling platform, which was funded, which connects infrastructure within UCD, the College of Surgeons, TU Dublin and UL, but also it's a national facility for access. We also identified, obviously, the North-South Research Programme as a key opportunity, and we had a webinar back in August for that particular programme. And we actually presented to the Joint Committee for Implementation of the Good Friday Agreement in October, and we were very well received. The other, I suppose, key thing that happened over the last couple of weeks is that we got the outcome from the North-South Research Programme, and I have a slide on that, but it was very successful. We had a big programme funded for AICRI for a postdoc PhD training programme, but also we had other areas funded. And the first workshop that we held was just in November last year on living with and beyond cancer. And this was essentially, I suppose, targeted in the area of the survivorship space. This was very successful. We had close to 100 participants. So again, the workshop that's taking place today is part of a series of 4 workshops. The next one actually is taking place in April. It's in the area of digital health, And then we have a last workshop on precision oncology and functional genomics. And what we're hoping to achieve from each of those workshops is a report where we kind of define what's the current state, what are the opportunities and how do we push forward. So I suppose this is the focus today really on these particular areas. And again, I'd like to thank the organisers for this event. And this is just a kind of an early highlight of the next workshop, which is taking place. It's a hybrid workshop, so there's in-person and online in Limerick, which is kind of co-organised with Dell. And this has been organised by Aidan Culhan and Mark Loader. Sorry, Isabel, am I still okay? Because I think Juriska is frozen there on my screen, so I don't know if I'm still projecting okay.
Yeah, no, it's still good.
Okay, great. So the IRC New Foundations Award, again, it runs up to August. This will provide funding for an AICRI website and then 4 workshops, the first of which is taking place next week, the Patient Stakeholder Workshop. And we have a Cancer Charity and Industry Workshop taking place a couple of months later. And then we have to do some work around kind of, I suppose, governance models for AICRI. And then the last point in the calendar is an AICRI workshop, which is taking place adjacent to the meeting that Professor John Crown organises, the translational meeting on oncology, which is happening in late September. And so we have a workshop in the morning of the 28th of September. So just to highlight that we have this patient stakeholder workshop, which is taking place next week in as an aside to the Irish Association for Cancer Research meeting. Again, I would encourage people to circulate that amongst any of their, if they have any connections within the patient advocacy community. We have already good participation. It's a hybrid event, so anyone probably in the Munster area, if they can make it, would be great, and that's fantastic. And just to finish off, I suppose, just want to highlight some recent successes. So again, there was a competitive call under the North-South Research Programme across all aspects of science and humanities. And there was a number of programs which were put in under either directly under the AICRI banner or kind of aligned to AICRI. Again, one program which I led in conjunction with other colleagues around Ireland was a postdoc PhD training program in the area of precision cancer medicine. That was funded. It was one of the 2 partnerships of scale programs funded. There was also 2 other successes which are quite relevant to AICRI. One, a program led by Professor Lorena Driscoll, and involving Paul Mullen in Queen's. This was funded in the area of liquid biopsies. And another program which was funded under the Strand 2 program was in the area of cancer data, which is a kind of key area of interest for AICRI. This was led by Professor Aidan Culhan and Mark Loader. And there were several other programs which were in the Strand 1 category, which were funded in this cancer space. So out of the approximately 37 million which was awarded, at least a third of the funding went to All Ireland or kind of interinstitutional cancer research programs. And again, this is just a slide to summarise a program which will be kicking off and encourage people to apply for this. Again, we're spreading it quite widely. There's going to be 10 PhD students across 10 institutions and 10 postdoctoral fellows, but we're hoping to grow that kind of program, use this as a model for a interinstitutional PhD and postdoc training program across all Ireland. I won't dwell on this, but obviously Juriska and others in the clinical sphere are involved in clinical trial activity, and again that has received recent investment from the HRB. A number of different institutions have been awarded funding in that area. And just to finish off, I know we're going to have a discussion later about, how do we actually fund AICRI going forward. So again, we have standard targets of SFI Research Centre programs. Also, the North-South research program is, will be funded or hopefully funded again. There'll be other funding in that area. But also the Peace Plus program is something that we're targeting as an opportunity to kind of bring people together. So in the interest of time, I'll finish off and hope to enjoy your discussions today. And I'm just going to stop presenting. So, Juriska, you're back up.
Thanks, Liam. That was a super overview of AICRI. And now we're going to start into the first of our 2 sessions for today. So today's workshop is going to be split into an immuno-oncology session where some of the basic science, translational and clinical researchers across Ireland who are working in the space will give us a 3 to 5 slide elevator pitch of their focus of research, and how this may be integrated into the cohesive whole of this pillar. So I'd love to introduce our speakers and ask them to give a brief one-line introduction and then to go straight into their elevator pitches. So our first speaker is Professor Aideen Ryan from Galway University Hospital and University Hospital Galway. Thank you very much, and I'll hand it over to you.
Perfect. Can you just let me know if you can see my screen and can hear me okay?
Yes, perfect.
Yeah, all good. Great. So thanks for the opportunity to present what my group works on. For those of you who don't know me, I'm a PI in NUI Galway with the focus, sorry, let me just see, my slides aren't progressing here. Can you see that okay? Yeah. So my background is really, I did my PhD in UCC with Professor Fergus Shanahan. And I followed that with a postdoc in tumour immunology in Galway, where I developed lots of animal models in colorectal cancer. Then moved on to develop stromal cell immunology expertise. And as you can see there, I'm not going to go through that timeline, but basically you can see that I've started my independent research group in 2017 at NUI Galway, where the main focus is on understanding how to use immunotherapies effectively in colorectal cancer and in other tumours. So just to give you a sense of the type of research we do, our publications are in three kind of areas: stromal cell immunology, basic mechanisms of action, cancer immunology, and then the intersection, which is the majority of the focus of my research at the minute, which is the role of stromal cells in the tumour microenvironment. And I'm fortunate to have a very extensive collaborative network, which obviously is part of this All Ireland Cancer Institute as well, could be extended, and I look forward to interactions with others. But we have a wide network of collaborators, both nationally and internationally, and they span kind of the academic and industry spaces. And just to give you an overview of how our funding looks, is I have funding from both national and international sources, and a substantial amount from industry as well, and I'll talk about that in the next couple of slides. So the premise behind most of our research is the idea that immunotherapy is the holy grail of cancer therapies and the potential that that has. However, as many of you will know, if you look at this graph, the response rates vary, and that's shown here on the y-axis. So you can have colorectal cancer that responds really well to immunotherapy and those that do not respond very well to immunotherapy. And there's a multitude of reasons for this, some of which relate to the mutational burden, which is shown across here on the x-axis. And the ones that respond better have higher mutational burden, but actually the ones that have lower mutational burden, there's a lot of different reasons why they don't respond. And that's because factors influence the response in the microenvironment. And this is where the majority of our research lies, in understanding how that microenvironment can shape immune responses. So there's two areas that I'm going to tell you about. One is novel targets of immunosuppression in the TME and how we go about that. So we know that stromal cells in the tumour microenvironment in colorectal cancer are associated with poor outcome. And that's in relation to their immunosuppressive abilities, but also how they regulate inflammation. And for those of you who are interested, I've just put an overview of the type of experimental approaches that we use to address and understand why these stromal cells are associated with poor prognosis in colorectal cancer. They include biobanking samples. We have a fairly extensive program on 3D model development, and also we can target immunosuppression using multiple different ex vivo assays. And the types of publications, for those of you who might be interested in collaborating, are just highlighted there on the right. The second area that we're interested in is identifying new ways to activate currently available immunotherapies. One of those approaches uses an immune-stimulating drug called cyclophosphamide, and in collaboration with both Professor Michael O'Dwyer and Dr. Janusz Krakow, we have established a clinical trial analysis platform where we link in with the clinical trial unit to assess both PBMCs and bone marrow of, in this case, multiple myeloma patients. And that involves immune phenotyping and functional assays using some of the technologies that are shown here on the right for us to be able to evaluate biomarkers of immune responses in those patients. And some of those publications that have been established out of that are shown here on the right again, for those of you who are interested. So to finish off then, I just want to give an overview of some of the research core facilities that might be of interest to this group in terms of infrastructure at NUI Galway that we use in particular. I have research focused in the Biomedical Sciences building, and also my group is located in the Lamb Institute for Translational Research, and that obviously has a lot of different advantages for access to direct access to clinical and patient clinics. In the Biomedical Sciences building, I've listed here, I'm not going to go through all of them, the different core facilities that are available, and also those in the Lamb Institute. So I'll leave it there, and if anybody has any questions, I'm more than happy to answer. And if anybody wants to reach out, I'm sure you'll be able to get my email address, or I can post that in the chat afterwards. Thanks for your time.
Thanks, Aidan, for that super presentation. I think what we'll do is we'll keep all the questions to the end and have a lively panel discussion where people can ask questions of each other and even to the speakers from the attendees. So I might move on to the next speaker, if that's okay. Elizabeth Ryan, who will be joining us from the University of Limerick and is going to talk about tumor-resistant myeloid cells and innate immune signaling.
Right, so just give me 1 second till I get myself sorted here. So can you see everything? Perfect. So okay, so I'm first of all going to give you a little bit about my background. So I graduated my PhD in vaccine immunology 20 years ago. And since then, I've worked in a diverse number of areas, studying different immune cells in academia, in industry, and in different contexts, both in infection, transplantation, autoimmunity, and in cancer. But the common theme really through all of my different research endeavors through the years has been really looking at immune cells at the interface, be it in the gut or the lung, and how monocytes within tissue interact with their environment. So it's a short presentation today, so I'm just going to give you a snapshot of some of the ideas and avenues that I'm establishing at the University of Limerick, where I moved in 2018, which seems like about 5 minutes ago because of everything that's happened recently. So I'll just start. So I worked before coming here to UL, I worked for a long time with the Centre for Colorectal Disease and collaborated very closely with Professor Glenn Doherty. And there we were looking at patients with inflammatory bowel disease and looking at their immune dysregulation, and particularly interested in why patients fail anti-TNF therapy. And this is actually really important. Some of the mechanisms underpinning this treatment failure in IBD can be really related to why patients on immune therapies develop side effects such as gut inflammation. And we've got a lot of information here that I can discuss with you in more detail at another time, but this is one area that I'm really interested in developing, and it's a very neat interaction between, autoimmune disease and immune dysregulation and the effects of cancer immune therapies. Another area at this interface between the microbiome and immunity are the way that the gut immune system interacts with microbial metabolites. We know from studying biopsies of colorectal cancer tissue that you can have colorectal tumors with very different metabolic environments. This is indicated here by the immunohistochemistry of markers of glycolysis or aerobic respiration. So you can have very different tumor microenvironments. This really correlates very strongly with the infiltrate of immune cells into that tissue. So understanding the metabolic environment of the tissue, how microbes interact with that, and how that impacts on immune cell function. Here we can see that butyrate inhibits the mTOR signaling pathway. And we've been doing some shotgun proteomics on cells treated with different microbial metabolites to really get to grips with the fundamental mechanisms underpinning this. And this will give us insight into this talk and this connection between the microbiome and the immune system. Now, something you probably didn't expect to see here was, looking in different animal models. And when you think about the innate immune system, mice are actually a really poor model for humans in many ways. And one thing that kind of was pivotal in my research career was when I was in Sanofi Pasteur. I was working with the vaccine formulation team, trying all different kinds of vaccine adjuvants to activate human immunity. And everything that would work in our animal models, nothing would translate in phase 1 clinical trials. However, I have been collaborating with a group in the University of Florida that study a model of turtle cancer called fibromyalitosis, which is a tumor that's caused by a gamma herpes virus that's very similar to EBV. So this drives this turtle tumor, and we have sequenced these tumors. And one of the single greatest predictor as to whether these tumors will recover and be rehabilitated and released into the wild or euthanized is their expression of these molecules that detect viruses. So these innate immune sensors that detect viral DNA, where they are downregulated or the virus has manipulated these to lower their expression, the turtles do really poorly. And we've translated some of these findings across into human colorectal cancer, where you can see that this tumors also vary in their level of viral sensors. And this immunohistochemistry here is for a protein called KHSRP, which regulates viral sensing through the RIG-I pathway. And I'm really interested in figuring out how latent viral infection manipulates viral sensors of viruses to impact on the immune response in tumors, because I think this is another, often overlooked way at which we get this variation in individuals to the immune infiltrate, what dictates the variation of immune infiltrate in different individuals. And the last thing that I'm quite interested in following up with this cohort is looking at dendritic cell markers and the activation in patients receiving immunotherapy. So 20 years ago, I published this paper about this dendritic cell molecule, and it was a difficult molecule to study, and we still really don't know what it means. But I've been recently getting an awful lot of people contacting me because it's coming up when people do these models looking for prognostic markers to checkpoint inhibitor responses. Here it pops up. It's renamed as CLEC-L1. It's a marker of response to PD-1, but I've also had some researchers from Rockefeller in New York contact me because it's also a marker to response to CAR T-cell therapy. I'm not sure this is mediated through the function of this molecule, but it's associated somehow with the response. And what's interesting is we found this molecule originally because it was upregulated by CD40. So I'm really interested in looking at anti-CD40 responses and that signaling pathway in patients receiving either CAR T-cell therapies or immune checkpoint inhibitors. So to finish off, I'm based in the University of Limerick. We have a huge amount of resources and facilities here that we are herding together to try and address some cancer-specific questions. If you go to the website, the Brunel Institute here has all kinds of kit. There's hundreds of millions of euro worth of mass specs and electron microscopy. We have also some spatial transcriptomics and multiplex immunofluorescence tools. We also work with the Health Research Institute, and we have a centre within the hospital in the UHL. And there we are supported with, the ability to do translational research. And I've started an immune profiling study there with Professor Ruth Clifford, where we're looking at patients with CLL and we're looking at their immune responses, their innate immune profiles, and their ability to respond to vaccination. So with that, I just want to thank you very much for listening to you and listening to some of my ideas, and I'm very much looking forward to working with this group.
Thanks so much, Elizabeth, for that great overview. From here, our next speaker is going to be Claire Gardner from the Trinity St. James's Cancer Institute and Trinity College Dublin, who's going to talk to us about immune cell therapies for cancer. So a broad overview. Claire.
Okay, hopefully my slides will appear. Can you see that clearly again? Yeah?
Perfect, yes.
Thank you. So just briefly to introduce myself, my name is Claire Gardner. I'm a professor in immunology, and I'm beginning to feel my age because it's actually 30 years since I graduated from college, and I've been working on NK cells ever since. I'm based in TBSI in Pearse Street in the Department of Biochemistry and Immunology. And I'm a research team lead for cancer immunology and immunotherapy, along with Joanne Lysett in the Trinity St. James's Cancer Institute. And again, I think AICRI is a very positive thing, and we're really looking forward to being part of this. So in terms of immunology, I guess I don't really need to introduce to this group how important it is, but I would like to give a little push on behalf of the basic scientific research and how that can impact in terms of downstream immunotherapies. So we know that the immune system protects against cancer, and that's what it's designed to do. And when that fails, it can actually cause cancer to progress. And that's kind of an area that a lot of some of the basic scientists that are working in this area are focused on. And immunotherapies, we all know, are a basic pillar at this point in terms of treatments. And we have, again, several ideas stemming from our basic work in terms of how we can improve current immunotherapies and to indeed develop new ones. So my pitch for immunology is that I think it is there's a great opportunity, not just specifically within the area of immunotherapy or immuno-oncology, but to really develop collaborations with basic cancer scientists. So when you have a cancer scientist who's interested in their particular cancer, the immunology can actually be applied to any particular cancer and is relevant in that concept, in that sphere, and provides a unique selling point, I think, for Ireland in terms of moving forward. So that rather than just have silos of cancer researchers and immuno-oncology, that we really bring in parallel these two strands forward to generate synergy. So in terms of what I work on an immune cell, a natural killer cell, and actually NK cell therapies aren't anything new. So there are lots of therapies, including rituximab, anti-GD2, trastuzumab, and so on, that have been around for a very long time for treating, lymphomas, neuroblastoma, breast cancer, and so on. And at least part of the mechanism of action of these particular drugs, antibody therapies, has been to harness the immune system to actually kill the tumor cell. So the antibody will localize to the tumor cell, and NK cells can come along and specifically recognize those tumors and kill them. So that's been known for a very, very long time. And one of the things we're looking at is how we can improve aspects of NK cells to improve this. So also in terms of the checkpoint inhibitors, there's obviously a huge amount of information out there, and they've been hugely successful. But it's probably less appreciated is that at least part of their mechanisms of action is through the impact that they have on NK cells. And in fact, there are a huge number of checkpoint inhibitors that are specific to NK cells that are only beginning to come through into clinical trials and so on. For example, TIGIT is now starting clinical, there are trials proposed for starting fairly soon in terms of TIGIT. But what I really want to talk about, because this is more where my immediate research has been focused, is in the area of NK cell therapies and how we can use them. So, and a lot of it has been focused and still is focused in terms of the autologous therapies, where you take NK cells from individuals, and you find that in cancer patients, these aren't really working properly. And so what we've been involved in is trying to understand why these cells are not working properly and how we can either manipulate them ex vivo, pharmacologically, or genetically to then improve them when those cells go back into patients. But this work also has impact in terms of allogeneic cell therapies. And we all know that at the moment, this field is just mushrooming in terms of interest and companies and so on that are looking at it. So whether it's cells from healthy donors, from banks, NK cells derived from stem cells, cord blood, cell lines, everything is going in terms of developing cell therapies. And CAR-NK cell therapies are obviously very, very topical. And again, there's huge potential here for great research, because anything we discover from either the basic stuff or from these technologies here, anything we can do to improve a CAR-NK cell in vivo is going to have great impact for patients. So my specific research questions have been how to make a better NK cell therapy for cancer, and how to better prepare immune cells for surviving in the tumor microenvironment when they're administered into people. And in particular, in Trinity, some of myself and my collaborators, we work in the area of NK cell immunometabolism. So we have a lot of expertise in that area, trying to look at NK cell dysregulation of NK cell metabolism, mechanisms of action there, with a view to actually modulating that so that cells can behave better in vivo. And we have loads of targets that we've already identified and are currently working on. And all the work that we do is applicable to both autologous and allogeneic cancers or therapies. In terms of the cohorts, the cancer cohorts that I've been looking at, I've been really focused on breast cancer. We're working now on metastatic melanoma. We've ongoing work in pediatric cancers in the area of neuroblastoma. And really my main focus is on human immunology. We do have people in the Trinity St. James's Cancer Institute that have lots of expertise in murine models of these things as well. So from myself, immunology, NK cells, immunometabolism, and I'm very open to collaboration, and I hope that we can grow immunology, basic immunology alongside translational and clinical immunology. Okay, so I will leave it at that.
Thanks so much, Claire, for that great overview of your background and how things will fit into a national group. From here, I'd like to move on to our next speaker, who is Donal Brennan. Donal Brennan comes to us from UCD and is going to talk to us today about tumor immunology in gynecologic malignancies.
Sorry, thanks. I'm just having trouble getting my slides up here for some reason.
Claire, I think you need to stop sharing yours.
I asked it to stop sharing.
There you go.
There we
go. It's reluctant.
Here we go.
Are you okay, Donal? Are you able to share?
I think I am. 1 second now.
Okay, great. Perfect.
Sorry there.
Yeah, we can see your screen now.
Okay, can you see is that coming up?
Yes.
Yes, perfect.
Great, thanks. Hi, thanks for the opportunity to present. My name is Donal Brennan. I'm the chair of gynecological oncology at UCD, and I suppose our program is unusual in that our clinical program spans across 2 hospitals, the Mater and Vincent's, and has been quite active over the last number of years within the immuno-oncology sphere. To kind of cut to the chase, myself and Walter Kolsch lead Precision Oncology Ireland project in collaboration with AstraZeneca. It's focused on improving immune checkpoint inhibitor therapies with a specific focus on ovarian cancer. Ovarian cancer, as you know, has a very poor prognosis, and it should respond to immune checkpoint inhibitors, but really does not. So we've taken a approach of using both dry and wet lab approaches to try and understand this in more detail, using a very in-depth collaboration with AstraZeneca. So there's approximately 15 people working on this project. And I'll show you some of the data from that in a minute. But basically what we've taken is a dry approach to look at the signaling networks and try and map them out in RNA-seq data, particularly single cell data, but also then to use some proteomics to understand the downstream signaling effects of immune checkpoint inhibition, with the idea being that we can then tap into the extensive knowledge within Systems Biology Ireland from a mathematical modelling perspective to see can we identify synergies that we could use for combination therapy with ICIs and other drugs. And we've heard earlier about some of the potential synergies, particularly with regards to metabolic inhibitors. I think from a clinical perspective, a thing to be aware of from a gynae cancer perspective, particularly ovarian cancers, we have a lot of large biobanks and we have access to a lot of tumour. But this slide just shows very clearly that studying the tumour microenvironment in primary tumours really doesn't reflect what happens in the metastatic setting. As you can see in the pie charts here, this is work done by one of my MD students and one of our postdocs, Karen Slattery, who works with myself and Lydia Lynch, that really the CD8 positive T cell count in the ovary is much higher than in the matched omentum. And that's probably one of the reasons why we assumed that the ovarian cancer patients would respond well to immune checkpoint inhibitors, because most of the data that was produced on primary samples rather than on metastatic samples. And that's one of the areas that we spent a lot of time working on now. The difference between a matched metastasis within the same patient, and ultimately the TME seem to be very different. Our work with Walter and myself within the group is really focused on two areas. As I said, the first is using proteomics to map out downstream signaling effects of different immune checkpoint inhibitors. We have a particular interest in PD-1 and TIGIT, and we've got quite a lot of nice cell line models that we're using to map these out. This just shows some preliminary data on the right-hand side of some proteomics, where we pull down PD-1 using nivolumab and pembrolizumab, which obviously bind to the extracellular domain of PD-1, but we've also used a cell signaling antibody which binds to the intracellular domain of PD-1, and you can see that there's a very different protein profile that's exhibited on the proteomics. And fitting in with what people spoke about earlier, some of the differences appear to be the interaction between PD-1 and metabolic cascades, particularly glycolysis. And I think this shows the way that we can use this sort of technology to really start to understand the downstream effect of PD-1 inhibition, but also TIGIT and some of the newer anti-immune checkpoint inhibitors that are coming along. With regards to, this seems to have frozen. Can you see this?
Yes, we can see it. With
regards to the dry lab work, what we've done is we've basically worked with some publicly available single-cell RNA-seq data sets, but developed a pipeline where we can identify regulons or master transcription regulators to really reduce the dimensionality of single-cell RNA-seq data sets, because as you know, they can be quite shallow and can be difficult to reproduce. But interestingly, what we've been able to do is map these regulons onto specific cell types, as you can see in the bottom left of the screen here, demonstrating a number of regulons and master transcription regulators that are active in different cell types, and transfer this into bulk RNA-seq data, which we can then use to look at prognosis of patients treated with, in this case, melanoma patients treated with immune checkpoint inhibitors. And this sort of approach could be very helpful with regards to how we use publicly available data sets, particularly those that have been using bulk RNA-seq to look at pre and post immune checkpoint inhibition.
I think Donal may have frozen. Isabella, any thoughts? I can turn my camera off if I'm the cause.
No, I think it's on his side.
Okay.
He's still logged in, so it might be his connection.
Okay.
Might need to move on maybe to the next one while he
Reconnects.
reconnects. Yeah, maybe. I don't know if you can hear, but it might be worth to reconnect.
I can send him a email. Oh.
Yeah, he's reconnecting, I think.
Okay. Okay.
I'll just take the chance to just remind everyone, all the attendees, to pop your questions in the chat. So for the discussions.
Okay. Will we give him a minute to reconnect so that he can finish? We're doing very well for time, actually. We're exactly on time. I'll give it another minute, and if he hasn't reconnected, then we can move on to Austin. Okay. In the interest of time, Austin, is it okay if you share your screen?
Yeah, absolutely. Yeah, let me.
Okay, so we'll return to Donald Brennan when he's had a chance to reconnect. Our next speaker is Professor Austin Duffy from UCD and the Marshall Hospital, who's going to speak to us about early phase cancer immunotherapy clinical trials.
Can you see my screen, my slides?
Yes.
Okay, great. Thank you, Yuriska, for the invitation to speak, and also congratulations, Liam and the group, on the wider AICRI effort. I think it's just brilliant, and very excited to be involved, and in any way really. So I'm going to talk about early phase clinical trials, which comes at this sort of from the other angle, which is what I'm hoping to do is establish a platform that other folks can plug into and sort of get at that interface with the patient, which is what I'm most interested in. And I will also, as you can see in the top right-hand corner of the slide here, I also want to introduce an initiative that we've been working on here at the Marshall for the last couple of years, and which is at fairly advanced stages in the development, and hopefully will become a reality in the not too distant future. Okay. So when we look at the stages of drug development, it's a very long and tortuous process, as Jeruska well knows, and it begins obviously back in the lab with our basic scientist colleagues and their scientific expertise and understanding, which is sort of the foundation for everything, obviously. And then there's sort of the drug, the candidate drug, let's assuming it does eventually make it to the market. It goes through this very sort of convoluted and rather tedious and difficult and challenging and long developmental story, beginning with all of the preclinical work and toxicology and so forth, before crossing that bright red line that I've got on my screen there and entering into the clinic, where patients get this drug for the first time in phase 1 or early phase studies. And you know, at that point, you're looking at toxicity primarily and safety. You're trying to figure out the dose and scheduling. You're getting to know the drug, and you're looking, of course, obviously for hints that it's effective. And then you take those hints, you move into the phase 2 setting. That's another few years down the line before eventually the drug is looking promising enough to be developed on a more global scale in the context of a phase 3 and potentially practice-changing study and making it onto the market. So this is a very long process, and I think and it's it's necessary. You know, there are certainly inefficiencies in it, but it's a well-trod path. Now, in Ireland, of course, you know, and this is a generalization, but most of the sort of clinical trials and research drug development space that we're involved in here is at that kind of later end, you know, in the sort of more phase 3 setting. Now, I know there are several earlier phase studies here and there, and some great research being done. But I think it's fair enough to generalize that in general, most of the sort of clinical trials and the drug development that's been done is at that latter end. And this is really all thanks, or predominantly thanks, to the great work that's been done by Cancer Trials Ireland, that many of the clinicians such as myself and Jerushka and others on the call, and translational folks as well are involved in. And this is really a great organization that provides Irish patients with access to drugs that aren't yet on the market. And if we were to sort of, again, do a further generalization by looking at this and this sort of milieu of how patients in this country get experimental drugs and get access to non-market approved drugs, but ones that look promising, we really are grateful to the work that's being done by many folks in this sphere. And Cancer Trials Ireland is a wonderful platform for interaction and engagement and dissemination and patient recruitment. There's a great sense of community amongst us all, even though we are siloed to a certain extent within each individual hospitals. We all meet up regularly. We're having our spring big meeting tomorrow, and so there's very much a network approach to the clinical trials that are being done. And this is kind of a necessity, I suppose, when you look at how we're structured and the need for geographical equity, making sure that patients in Cork, for instance, can access clinical trials just as much as patients in Dublin or Galway or so forth. So the approach is kind of networked and distributed, and these are not negative things, but I would say that probably does impact on the type of research that we do. So, when you think about it, if you have a sort of this networked approach, which is absolutely a positive, it does mean that you're kind of automatically in that later phase territory, right? Because it's harder to do sort of more early phase trials if, in multiple locations, just for the sheer practicality of it. And then late phase, this has a knock-on effect that the late phase studies that you're kind of doing, tend to be very disease specific. Yes, there are some basket studies out there, but they tend to be disease specific. It tends to be an international, cooperative effort. Again, that's not a bad thing. It's great to be plugged into these international networks. So I'm in no way being critical of that. And the trials, these sort of large scale phase 3 trials tend to be, not always, but tend to be in the first line or in the adjuvant setting, in order to get that practice-changing purchase on the market that our industry partners, are so hopeful for. So all of these are good things, right? And they certainly, as I said, they provide access to Irish patients for non-market approved drugs, and it works very well for the most part. But I guess if you were to flip it around, and I think you could make the argument that there is a much needed space there for almost the opposite approach, where instead of this wonderful networked approach that we can all plug into, where actually you don't strive for that so much as you turn it on its head and say, well, why don't we have a dedicated space, that's focused purely on earlier phase research. And let's look at the advantage of those, which I have listed on the screen. Well, if you have if you shift or include more space for earlier access in that, drug development journey I showed you a couple of slides, there's a few advantages to that for our patients. First of all, earlier phase trials tend to have broader tissue eligibility. A lot of them are tissue agnostic, and you just require a patient to be fit and well and have good lab results and that sort of thing. So therefore, it also caters for rare and uncommon cancers, not even rare cancers, but lesser common major malignancies. It addresses the needs of a very prevalent patient population, which is those with metastatic disease who have already been through a couple of lines of chemotherapy. And it's very hard to get a phase 3 study for these patients. It gives you access to novel drugs, almost by definition, right? It's an early phase unit. These are drugs that have just come hot off the press from our bench collaborators, you know, and are ready to enter the patient setting. So it brings our patients earlier on in that sort of drug development journey. It also provides opportunities for critical mass accrual within one site, and you can have spin-off studies, you know, looking at different aspects of this novel drug. And it also has knock-on effects for training and expertise development and grants and so forth. The main challenge, of course, is to retain that wonderful network approach that we already have, and to embed it within the great work that Cancer Trials Ireland do. And I'm talking about engagement here that's just beyond looking for patient referrals. So a lot of these advantages of an early phase unit, I believe, can really boost our patient participation rate in clinical trials. And that's, we know, a key metric that the HRB and other folks we're all looking at, right? We're all trying to increase participation. Just a very quick point that in the UK, you'll see this slide, there are 18 dedicated early phase clinical trial centres in the UK, including Belfast. And these are just the ones that are totally affiliated with this Cancer Research UK. And notice that in south of the border in Ireland, we unfortunately don't have any footprint there. So I just want to mention this initiative that I highlighted at the beginning. It's not fully over the line yet, but again, we're hoping to bring this to Dublin. And this is an organization called START. What are START? They are a research group that was founded by a group of oncologists, basically in San Antonio, almost 20 years ago now. And they got together and they basically developed a standardized, this is key, standardized method of doing phase 1 trials, with a very streamlined, efficient process that they then could offer to industry partners. And they quickly grew to become one of the largest, if not the largest, phase 1 research group in the world. They've got a very high track record. They developed the first in human study for pembrolizumab, for example, other drugs such as bortezomib or carfilzomib, several other drugs that we all are familiar with. Also have a very active xenograft program and a good publication track record. And we're hoping to bring them to the Mater and open a collaborative facility here in partnership with them that we would very much want to make a national effort beyond the Mater. And this is why this initiative is such an important thing that we can look at going forward. But my hopes for this venture is that, you know, by partnering with these experts, these world experts in this particular space, we can learn, we can train our staff and our junior investigators to really become leaders in this particular area. And this is all, again, with standardized uniform SOPs, which would make life so much easier. And it can also, and this is particular to this group, act as a platform for academic engagement, spin-off studies, working with some of the investigators who've already spoken and who I'm sure would be very interested in some of the novel drugs and agents we can access through this program. But most importantly, of course, it complements the great work that Cancer Trials Ireland does, and increases, and this is really the key for me, it increases the trial options that are available for Irish patients with advanced cancers. So I'm going to stop talking there, and we'll be happy to answer any questions later on in the discussion part of it. So thank you, Jerushka.
No problem. Thanks, Austin. Great initiative, and we're really looking forward to being able to contribute to that. So Donal, I see you're back on. We'll come back to you at the end if that's okay. I might go to the end and to the end of Joanne's, and then we'll come back to your presentation. Can everybody see my slides?
Yeah, we see fine.
Perfect. Okay, so I'm going to talk a little bit about a project that I've been working on with several investigators on this call, and some background for myself. So the project is a national immunotherapy biomarker and toxicity protocol. So as some of you may know, I am an unlikely Irish. So I'm South African, but I trained in Trinity and did all of my training in St. James's Hospital, followed by 1 year in Beaumont. And then went to Memorial Sloan Kettering, where I specialized in lung cancer and phase 1 immunotherapy clinical trials with Jed Walchok. After that, I spent several years at Johns Hopkins University as an assistant professor in the lung cancer division, where I developed a specific focus on biostatistics and data science, clinical trials, including investigator-initiated clinical trials, and immune-related toxicity. So during my time there, this is an example of an investigator-initiated clinical trial I wrote, funded, and was recently published, investigating the potential utility of anti-PD-1 immunotherapy in patients with metastatic disease to the brain, identifying a response rate of nearly 40% for intracranial tumors with immunotherapy, and also identifying biomarkers of response in the CSF. And we were able to present this as a late-breaking oral abstract at the SITC meeting, and it has subsequently been published in the Journal of Immunotherapy for Cancer. From a data science and biomarker perspective, obviously I was very lucky to be steeped in the culture of Sloan Kettering and Hopkins, and did some work on PD-L1 expression in rarer thoracic tumors. And then, of course, hence the involvement in this group, collaborated with several gut microbiome specialists at Memorial Sloan Kettering, Eric Paymer, and at Hopkins, Cindy Sears. And we won a grant to identify microbial biomarkers of response to toxicity in patients with lung cancer, and accrued 200 patients in 2 years. And we are currently analyzing the data as part of an ISLAC Career Development Award. From a data science perspective, it became very clear to me very early on that as part of team science, working with basic scientists, translational researchers, and sort of leveraging the skill set of data science, we can identify biomarkers that may inform investigator-initiated clinical trials. So in order to do this, at Hopkins, we set up a biorepository and an accompanying clinical database where every single patient with thoracic malignancies had baseline and on-treatment blood samples that were collected. And this is an example of our sample repository. So you can see nearly 10,000 samples that were collected over a 5-year period across different tumor types. I led the development of the clinical database, comprehensively annotating all of the samples, and helping basic science researchers and ourselves come up with interesting clinical insights to inform clinical trials. And these are some of the projects that we were able to publish. So this paper in Nature Cancer, led by my colleague Elsa Anagnostou, in which she developed a multi-omic predictor of IO response, integrating tumor mutational burden, but also bringing tumor purity, RTK mutations, a smoking signature, and HLA variation into a Cox model. And then a paper that I led, looking at the incidence of immune-related toxicities and the association with response to immunotherapy in patients with lung cancer. And this data published in JAMA Oncology recently, which demonstrates that with each incremental immune-related toxicity a patient develops, there is a statistically significant association with response while controlling for immortal time bias. My third area of deep love and interest is in the mechanisms and management of immune toxicity. So I set up a multidisciplinary team at Hopkins, which started really small, around 6 individuals, but expanded to about 20 individuals by the time I moved home, in which we had a really dynamic electronic referral system where patients with complex immune toxicities could access an immune toxicity expert group, and we were also able to collect clinical data and samples as part of our original database. So bringing this home to Ireland, as part of the AICRI proposal, we put together an idea for a very similar biomarker collection protocol where we will obtain blood and microbiome samples at serial time points in patients with tumor types that are treated with immunotherapy. This is currently approved in Beaumont, and the goal is to open it starting between Beaumont and Cork, and to enroll just over 200 patients over the next couple of years. Of course, we would love to utilize this as a platform to open nationally, and more than interested to hear from others on this call and more broadly who may want to be involved. Another hat that I wear is obviously clinical trials, and as a medical oncologist and clinical trialist, this is one of my #1 loves. So I'm the lung cancer lead for Cancer Trials Ireland, which Austin just mentioned. This is our current clinical portfolio, and since I came home in the last year and a half, we've gone from 3 trials to 9. So we are expanding our clinical trial portfolio. And then to try to foster homegrown science and investigator-initiated clinical trials, we've developed the Irish Lung Cancer Alliance, which is a group that started again with 30 individuals and now has 49 individuals from different institutions across the country who meet every 6 to 8 weeks on Zoom meetings, similar to this, share our experience. And so far, we have 1 investigator-initiated clinical trial that has been written and funded through a European group. To bring the toxicity experience forward, I'm working with nurses and patient advocates to develop a similar electronic referral system, working not just on IR toxicity, which will be supported through an MDT, but also open questions such as, now that we're treating more and more patients with immunotherapy, we are seeing survivors from immunotherapy, and the biomarkers associated with long-term toxicity are largely unknown. So we hope to create an IO survivor clinic to try to understand some of these questions further. So next steps for this protocol, it's currently ethics approved in Beaumont. We're currently going through the process of material transfer agreements across the different institutions. Many of the investigators on this call are involved, but of course, we're more than happy to hear about others who may want to be involved. I'm bringing a toxicity team together to independently assess the tox events, and of course, hoping that AICRI and other similar platforms may help us to fund ambitious projects such as this. Okay, so I'm going to move on, if that's okay, to Joanne. So Joanne Lysett is an associate professor of immunology in Trinity, and she's going to give us our next presentation before Donal finishes his.
Great, can you see that okay?
Perfect.
Great. Thanks, Drushka, and thanks, Isabella, and everyone who organised this great workshop. I think it's going to be really important, and just delighted to be part of AICRI and see where it's going to go. So, as requested, I'm just going to give a very brief background into my research track record, and I just want to apologise for my croaky voice. I've avoided COVID until now, but it's going to catch up with me at some stage. So I did my undergrad in biology and maths in Maynooth University, and I did my PhD in tumour immunology in Trinity with Professor Kingston Mills. I was appointed an assistant professor in 2011 in Trinity and promoted to associate professor in 2017. So I've established and lead the translational research group in Trinity called the Cancer Immunology and Immunotherapy Group. I currently, that group is made up of 1 senior postdoc, 8 PhDs, 1 MD and 3 master's students. And this is just an example of some of the funders that we have currently for our ongoing research. And some of the other roles that I have that may be relevant to this group is I'm the Cancer Immunology and Immunotherapy Research Team co-lead with Claire Gardner for the Trinity St James's Cancer Institute. I'm the Secretary of the Irish Society of Immunology and a Senior Council Member for the Irish Association for Cancer Research. So my area of expertise, I suppose, is really translational research on T cells and anti-tumour immunity, but also targeting T cells with immunotherapy. And obviously to do this work, we need a lot of national and international and academic collaborators. And here's an example of some of the institutes and industries that we work with. So in terms of the cancer types that we focus on, predominantly my group focuses on esophageal adenocarcinoma, but we also look at other upper GI cancers, including gastroesophageal junctional cancer and gastric cancers. And you see the 5-year survival rate for OAC is about 20%, and numbers have tipped 500 per annum. We also have projects in pancreatic cancer, again, a very dismal 5-year survival rate of about 9%. And we just recently started working in ovarian cancer. And again, the survival rates are pretty poor. So the cancer types we work on are pretty dismal outcome cancers. And we have collaborators with colleagues across multiple hospitals and also other institutions as well. And this is ongoing for all of these projects. So in terms of the research focus of my group, a high level kind of overview, it could be divided into two main areas. The main one would be really assessing the health status of T cells in cancer patients. So we're really interested in how the microenvironment affects the health status and the functional properties of T cells in particular. So how the primary tumor, the draining lymph node, and we're hoping to move into metastatic sites, as Donald mentioned, the importance of assessing, I suppose, the function and the health status of T cells at all these different sites and how they differ. So in terms of the tumor microenvironment, particularly solid tumor, there's a lot of physical features that can profoundly impact anti-tumor immune responses. So we're interested in nutrient deprivation, hypoxia, and acidosis. We're also very interested in the impact of the treatment that the patients are receiving anyway and how that's going to impact on T cell functional status. So for OAC in particular, it would be chemotherapy, radiotherapy, and surgery. And it's really important for us to understand how the TME and standard of care can impact T cell health status. So whether it's immune stimulatory versus suppressive, how this impacts their function status, the T cell metabolism. We also have programs looking at trafficking, so looking at blocking or targeting the chemokine system to see if we can recruit anti-tumor immune cells into the tumor site. And we also look at NK cells as well in this area. We look at the impact of, or the potential pathological role of T cells as well in carcinogenesis, particularly in inflammation. And in this case, we've studies in obesity and also cachexia. And obviously how all of these are going to impact on the response to immune checkpoint inhibitors. So for us, understanding all of these different factors are really important, and they're crucial information really when you're going to select an effective immunotherapy, and if you want to try and modify or improve the response rates to immunotherapy. So the other half of the research really is more novel, and it's really emerged over the last 4 to 5 years. And it's really looking at the impact of immune checkpoint inhibitors on the tumor cells themselves. So these are immune independent effects or almost off-target effects of immune checkpoint inhibitors. So we know that tumor microenvironment and standard of care treatments will affect immune checkpoint receptors on the T cell themselves. But we know this have started to show that you can have immune checkpoint receptor expression on the tumor cells themselves. Signaling through this immune checkpoint receptors on the tumor cells elicits a number of hallmarks of cancer. We've evidence to support intrinsic signaling through immune checkpoint receptors in promoting immune evasion, affects DNA repair, chemosensitivity, proliferation, metabolism, apoptosis, but also cancer stem cells and senescence as well. So again, knowing the health status of the T cells that you're trying to target with immunotherapy, but also the potential off-target effects of immune therapy is really important. So again, when you're trying to identify what's the most appropriate immunotherapy to use, also, it's important to understand when the best time is to incorporate immune checkpoints into standard of care treatments, be that in the adjuvant or neoadjuvant setting. And also, what's the best combination treatments? Is this with other immunotherapies or is it with other standard of care regimens? So in terms of core facilities at TTMI, we'd particularly, I'm just going to highlight the ones that we'd use. So we've hypoxia chambers, again, which is really important for our TME studies. We have an extral X-ray radiator, which is important when we're looking at mimicking standard of care treatments in the lab. And this is the only X-ray radiator for use in Ireland for research. We have a number of seahorse machines for real-time metabolic analysis, and one of these is housed in a hypoxia unit, so you can assess the impact of hypoxia on cellular metabolism. We have a flow cytometry and cell sorting unit. Obviously, biobanking is hugely important for translational research, and the upper GI biobank is almost 20 years old, and we biobank blood, serum, a whole range of different tissue types, both pre and post-treatment, and that's really crucial to our research. We have a number of different multiplex secretome analysis platforms, genomic analysis as well, which houses the BD Rhapsody, which Liz had mentioned was in Limerick as well, so for single cell seq. And we have a number of fluorescent imaging platforms as well for live cell culture. So as I mentioned, my group is based in TTMI, which is on the campus of St James's Hospital. So we have very close collaborations with clinical and allied healthcare professionals, which is hugely important both for translating our research and hopefully translating them into early phase clinical trials. And we form part of the Trinity St James's Cancer Institute, which is an OACI accredited cancer centre. So that's it. I'll leave it there. I'd just like to again thank the organisers for inviting me to speak here today, and I really look forward to the discussion.
That's great. Thanks so much, Joanne. Great overview. So I might just circle back to Donal.
I think, Juriska, in the interest of time, we might go on to the conversation, onto the discussion. If that's okay with you guys.
Oh yeah, that's perfect. So first of all, I'd like to thank all of the speakers for showing us their work. And I think, you know, the purpose of this meeting is to encourage cross-pollination of ideas and areas that we feel could be mutually supportive. So to kick off some of the questions, I think it would be useful to maybe go through the speakers and ask them, you know, if Carlsberg did collaboration, and there was one thing that you think would be helpful to your research and to IO research nationally, what would that be and why? Maybe I'll start with Joanne, since she was the last speaker.
Thanks, Juriska. I think for us as well, for translational researchers, is getting access to patient samples from patients who are on immunotherapy trials, or even that link for the next stage, getting our research to early phase clinical trials. And I know Austin has alluded to it as well, but I think it's a real deficit at the moment. And it is a real block. So I think there is that black hole of a lot of translational ideas, and basic and translational ideas, that just kind of get lost and don't get translated to the patient at all.
Yeah, I hear you. And I think, you know, I can go next since I was the speaker before you. This is sort of the thinking behind Irish Lung Cancer Alliance, or hopefully some of the working groups, is to sort of allow clinical folks and basic science to create that system. Donal, you've been very successful in doing this with Lydia so far. What's your secret sauce? Can you sort of maybe highlight what you think has really worked, or maybe what you think could be better?
Yeah, I think so. There's a couple of important issues. Getting, actually, as you know, getting access to patients in pharma-related trials can be very difficult because of all of the MTAs around that. So, but ultimately, I think it's really a lot of hard work on the clinical side to set up appropriately funded biobanks, and I think the HRB CRC clusters will add to that, not the trials clusters, but the actual clinical research centre clusters. Our biobank, like the one in Trinity, is 6 or 7 years old now. But the other big thing I would say is most of our sequencing is now done in Harvard, and there was a huge amount of work that had to go in moving tissue from the EU to the US. So I would think most of the work that needs to be done is actually at the governance level for biobanks. If you get that governance level right at the start, the rest of it is actually pretty straightforward.
And when you say get the governance level right, what do you mean by that?
Well, I think having the proper SOPs in place with regards to data transfer agreements, MTAs. Lots of people have biobanks, but when you try to share, particularly with pharma, you may not have consent from the patients to do so. Particularly in ovarian cancer, a lot of the patients are dead. Our biobank took about 18 months to go through ethics because of the fact that we needed prospective consent from the patients to do studies that they may not be able to consent to if they're dead. And if you don't have that in place, your biobank, it's like having a bottle of wine in a wine cellar that's open. It's not going to get any good. So that's the first thing. And the second thing then is, what's the relationship between the academic institution and who may well be in charge of the biobank and the hospital who's providing the actual samples? And there must be a very clear agreement in place between those for each biobank. And sometimes that can be very difficult to work through.
You're muted, Juriska.
Sorry now. Austin, your thoughts?
Yeah, so I think for me, you know, the thing, I mean, I've been back in Ireland about 4.5 years now or so, and the thing that, you know, has the most obvious thing that struck me almost straight away and drives me a little bit crazy is the fact that you've got so many great basic scientists, right, doing amazing work on this international level, and you know, we've seen the presentation so far, just really just brilliant. And then on the clinical end of things, you've got people like yourself, myself, Jerushka, and a lot of other folks, you know, spread around the country doing good, really working hard, trying to, you know, bring these newer drugs to the patients. And there's just it's like you've got these parallel tracks that are meeting maybe in infinity, but and yes, there are these cross points that folks have you know, highlighted, and you've just highlighted Donald's work with Lydia, and Aideen and Liz have also shown their work, and it's great, but it's so much it's so labor intensive to get that, particularly in the when you're dealing with investigational drugs, because you know, you can certainly set up a biobank, you know, to, you know, looking at surgical samples or taking bloods from patients going under standard of care. But, you know, my main interest is in, you know, investigational drugs that aren't standard of care. And for me, the nub of it comes down to, well, you know, the type of research, the type of clinical trials that you're doing. In other words, you know, what is the vessel that patients are in to access these investigational drugs, and then how do you link that to, you know, the bench? And it's a big problem because, you know, you've got, as I've said, you know, great work being done. I'm not being negative in any way, but it's a distributed sort of network, as is appropriate. I'm not arguing against that, but it does mean that you're drawn as a clinician towards taking part in late phase 3 trials that, you know, where the intellectual property, you know, is overseas in Boston or Berlin or wherever, and we don't have any input into the writing of the clinical trial protocol. We can't say, okay, we're going to give drug X on day 1, and I'm going to take a blood sample and I'm going to give it to Joanne, you know, on day 8 or something like that, so she can look into some novel mechanism. And even if we could do that, even if we could secure the sort of permission of the sponsor in the faraway land, you know, who are only dealing with you through a CRO, you know, even if you could secure that permission, you don't have critical mass patients, right? You just have a small numbers separated throughout the country. And so then you go, okay, well, what's the solution? Well, the solution is to do an investigator-initiated study, right, where you have total control, you write the protocol, you decide who you collaborate with. But, you know, and you know this just as well, if not better than me, Jerushka, and we've both been through this, and we're opening them, you know, I've opened a couple since I've been back. Each one is like starting a business. It's just, and listen, I'm no problem. I can spend my career opening investigation-initiated clinical trials. You know, if I hang around long enough, I might, you know, let's be optimistic. Let's say I open, I don't know, 5 or 6 of them, you know, that's fine, you know, but that doesn't change the systemic situation. You know, if you added up each, let's say, let's be optimistic again. Let's say each study I treat like 20 patients. At the end of that situation, you've got maybe you know, 80 to 100 patients over the span of decades who are accessing novel drugs in the context of a clinical trial where you can interact with this with all of the local academia. You know, and that's where I'm coming from in terms of we need a platform to allow us to do these sort of earlier phase studies, and where we have a little bit more control. I mean, yes, you still have to engage with, you know, you're working with industry who own the products, but I think that if you know, the early phase section, the early phase space for me, you know, is rich with opportunity for those sort of collaborations that don't really exist, or at least are very challenging to make a reality in the late phase setting. So that's kind of where I'm coming from. I know that was a very long-winded answer, and I apologize.
Okay, that's okay. No, very important insights. Just so that we're not running too crazy over time, I'll just ask Claire and Elizabeth to give brief comments each, if that's okay.
Thank you very much. I'd like to just echo what Austin said, and from the opposite perspective of an academic, we don't have the skills that we need to be able to take our ideas through to clinical trial. And I do feel like there is a missing link between enabling us or something to bring us through to the point where we have clinicians that we know are open and interested, and we can brainstorm, and they can tell us, well, actually, your idea is really good, but that's a problem there, and this is what you need to think about to bring that through to X, Y, or Z. So for me, it's a link there. I think, you know, easily in AICRI, we could have, off the top of my head, 20 good candidates for clinical trial, but we need more help.
Yeah. You need translators.
Yes.
Okay, we'll clone Austin and I.
Yeah, excellent, excellent.
And Donald's on the call as well. Elizabeth, any thoughts?
One thing, though, is from the researcher perspective, that could be improved is the career track for researchers. Because it is so precarious, it's very difficult to keep talent and to nurture and train people who have the skills to do this. You know, most people leave to actually do the job in industry because that's where I was really privileged. I was able to string together 18 years of 1- and 2-year contracts. That's really difficult. And we have such a leaky pipeline of talent, and that is something we need to get a career track for translational researchers to lead the biobanks and to collaborate and to bridge that gap.
That's a great idea, Elizabeth. Donald, your hand is up, and I might just have one question from the chat after that, and then we can close. Donald?
Yeah, sorry. I think the one thing that needs to be said is, and this is a common theme across all cancer research in Ireland, we're a tiny country, and maybe the best thing to do is to come together as a group and decide on a specific theme that you could actually work towards as a group, because everybody is doing great work, but everybody is quite separated and doing slightly different things. And, you know, that I think if we could come together as a group of researchers and clinician scientists and agree on broad themes that you want to work on within immuno-oncology, whether that's immunometabolism or even being more specific around certain areas like that, I think that's the only hope you have of actually getting to clinical trials, if that's, because otherwise everybody is doing a small piece in a very large jigsaw, and they're underfunded, and they're not actually going to get to where they want to get to.
That's a very good point. I see there are a couple of questions in the chat, but I'm cognizant of time. I think from here, I should hand over to Professor O'Toole and Professor Ghosh, who are going to talk to us about the microbiome, and hopefully we can come back to these questions at the very end. Thanks, everybody.
Thank you very much, Jeriska. And both Paul O'Toole and myself will chair this session. I will kick off with a little bit of a verbal introduction, and then I will hand over to Paul to take us through a slightly expanded intro that Paul will deliver, and then we move to the short presentations. We have a fewer number of presenters, and so we should be able to discuss a fair bit. So my name is Subrata Ghosh, and I moved to Ireland in April last year. I'm a gastroenterologist by training, and I had my first faculty position in University of Edinburgh, where I lived for about 10 years before taking up the chair of gastroenterology at Imperial College London at the Hammersmith Hospital. And then after another 8 or 9 years, I moved to Western Canada, University of Calgary, as the chairman of medicine and the head of department of medicine. And that's where I started to work a fair bit on inflammation-induced cancer, specifically with inflammatory bowel disease. And I am essentially focused on immunology of inflammation. But I've also developed a lot of drugs that are in play in IBD or up and coming in IBD over my many years. I returned to inaugurate an Institute of Translational Medicine in Birmingham. And that gave us a lot of opportunities, but that's where I got interested in checkpoint inhibitor induced adverse events, especially gut adverse events. And this is something that I intend to pursue. So I was fortunate to come to Cork. I always loved APC microbiome, and it's great to get an opportunity to work there. And I was even more fortunate to obtain a SFI research professorship. And one important element of that SFI research professorship, which is focused on immunotherapy of inflammation or chronic inflammatory diseases, is also to look at checkpoint inhibitor-induced toxicity, especially gut toxicity, partnering with Zerushka and, of course, partnering with Paul O'Toole. And I will finish by saying that this will be a great opportunity to make some of the elements of a checkpoint inhibitor, still resolving mysteries with a tremendous focus on gut microbiome, not only in kind of a modulating efficacy of checkpoint inhibitors, but also potentially predicting adverse events, and whether modulation of the microbiota can lead to enhanced efficacy. This is a common theme for chronic inflammatory disease immunotherapies as well. So my SFI research professorship, which is based on a program called Augment, is essentially looking at how we can use and override compensatory circuits, very often based in the gut microbiome, to make existing immunotherapies more effective. And as a platform, I just highlight that I was also fortunate through the SFI research professorship to obtain imaging mass cytometer, so we can do high multiplexed rare metal tagged antibody mapping of various cellular players in this whole process in spatial profiling. And this will be established, and then we need time to kind of really evolve our bioinformatics around it. But by next year, we'll have a good Hyperion imaging mass cytometry platform. I'll be using it a lot for looking at stromal cells in chronic inflammatory diseases, but I will welcome any efforts to look at stromal cells in the cancer tumor microenvironment as well. So with that, I will hand over to Paul, and Paul will take us through the rest.
Thank you very much, Subrata, and good morning, everybody. If you're worrying about the housekeeping, we will shorten the lunch break. You are probably going to eat it at your computer anyway, so you'll have even less time to check the news on RTE and read your lunch before we resume after lunch. I'm delighted to present an extended introduction into the topic of the microbiome and cancer. If you had told me 10 years ago that I'd be presenting at oncology workshops, I would say you're crazy. Even though I had worked on Helicobacter pylori and I was familiar with the notion that microbes could cause cancer, I was working on commensals. But as you probably know, and as I'll exemplify very briefly, we now know that the microbiome has a role in the causation of cancer and in the treatment of cancer. And as you'll see in a moment, we're also exploiting it as a diagnostic for certain cancers. And I work in the APC Microbiome Ireland, where we've been analysing the role of the microbiome as a risk factor for various diseases and in promoting health for the last 20 years or so. So the microbiota is a repository of extra genes which are involved in modulating aspects of host phenotype, including nutrition, immunity, and, as we now know, a risk factor for cancer. We've known for a long time that microbes like human papillomavirus and various other viruses can cause cancer, and HP, as I mentioned, is linked to gastric cancer. But what is new in the last decade is the realization that elements of the gut microbiota, which were assumed to be commensals, also modulate our risk for colon cancer through a variety of molecular mechanisms, which are to some extent works in progress. And in the context of this particular workshop, exciting work in the last decade, and indeed the last 5 years, has shown that the responsiveness of patients treated with immunotherapy depends to a large degree on the composition of their gut microbiome. And to make a long story short, a high diversity microbiome is typically associated with responsiveness, and a low diversity microbiome is the opposite. But there are few, if any, unifying taxa identified across different geographies, different cities, even in North America. So how are microbes involved in causing cancer? Well, this is some experimental evidence from preclinical models. It's an AOM DSS model, 3 repeated pulses of DSS. And the key thing is that in antibiotic treated animals, you don't see development of these tumors. And I teach genetics for a living, and I have to say I thought it was unlikely that it was worth investigating the microbiome of colon cancer biopsies for the presence of microbes which might have been involved in the driving or causation of this cancer. However, I was wrong, and in one of our first papers 5 years ago by a postdoc in my lab, Burkhard Flamer, we showed that the microbiome of colorectal cancer biopsies was different from that of polyps and that of controls. Being microbial ecologists, we were able to divide them into 7 ecological microbiome types, which we've since reduced to 5. But importantly, when we look for the presence of these clusters in other international data sets, we were able to find them undetected by the original authors. But this sort of underlying microbial ecology was present in all of the colon cancer associated microbiomes. And our vision for how microbes cause cancer is that a lot of these taxa are actually oral pathogens. And what we believe happens is that a colonic microbiota is either permissive or restrictive for allowing oral microbes to establish biofilms. You may have heard of Fusobacterium nucleatum, which is a well-characterized example, and either before or after predisposition key mutation, such as P53, the microbiome pushes it over the edge to form a tumor. The association of the oral microbiome was particularly interesting. This is a PCOA, which is not very sympathetic, but does illustrate that the oral microbiome of colon cancer patients is different from that of polyps or controls. And working with a campus company, which Fergus Shannon and I established, we were able to use simple machine learning and artificial intelligence to develop superior microbiome-based diagnostics for colon cancers and even for polyps, which are very sensitive and specific. And the rationale for combining the oral and fecal microbiome is resonant with the theory I presented a couple of minutes ago. We're interested in the patient journey and cancer survivorship. And these are the recently accepted results from a study we performed in collaboration with Eilish O'Connor in University of Limerick, because I don't know anything about nutrition. So the blue circle represents the microbiome of CRC patients, and the yellow circle in the background is actually non-CRC. So these are healthy controls. And we looked at the microbiome, the gut microbiome of post-op patients. We found that they fell into 2 groups, this sort of pinky purple color, which is a cancer-like microbiome being more related to the original cancer microbiome, and a green post-op normal type microbiome. So it seems that after the full patient journey of treatment and surgery, 2 different microbiome types are emerging where they either retain the risk-associated microbiome or they acquire a healthy one. And not surprisingly, one of the strongest metadata to associate with patients acquiring a normal-like microbiome was their habitual diet, including various kinds of vegetables. And we've previously seen that one of the strongest determinants of the microbiome of cancer patients at baseline is whether or not they consume various fruits and fibers. So we think that one of the classical Dennis Burkitt links to the cancer-causing microbiome is mediated through diet and leveraged through the microbiome. lastly, I want to briefly describe some work we've done with a number of people, including Clodagh Murphy, some people in my lab, Dr. Derval Collins, Professor Fergus Shannon, and Derek Power, on the microbiome-associated responsiveness and side effects with immunotherapy. So this was essentially a pilot study where we looked at the microbiome, which was clearly different between controls, responders and non-responders, and there were similar differences in the side effect occurrences or not. And these are some of the taxa which are associated with responsiveness and presence or absence of side effects. And some of these taxa are usual suspects like Akkermansia and Faecolobacterium prausnitzii, but others are not. So in the context of the discussion we're hopefully going to have about national initiatives, I think Ireland is a relatively small, relatively homogeneous country in terms of geography, ethnicity, and environmental factors. I think one of the things we can investigate productively are the environmental modulators of the role of the microbiome in immunotherapy responsiveness. So rather than hogging any more time, I want to hand over to our group of speakers, Andrew McDowell, Ken Nally, and Joachim Pren. And Andrew's going to lead off and present his research on the host microbiota and prostate cancer.
Thanks, Paul. I'm just saying about my presentation. I'm not sure if it's coming up.
Yeah, it's coming up now. Can you see it, everyone?
Yeah, that's great. Thank you. Okay, so firstly, can I say thank you for the invitation to speak this morning. So maybe start with just a very brief bio, just talk a little bit about my background, my research interests, and then talk a little bit about some of the cancer research that's taken place at Ulster. Focus on some of the work that we have been doing in relation to the human microbiota and cancer, primarily prostate cancer, and then finish just with a little bit of information on the infrastructure that we have at Ulster and some of the other resources that are available. So I'm currently lecturing molecular microbiology within the School of Biomedical Sciences at Ulster, although most of my research is focused in the area of medical microbiology. And I'm also the Associate Course Director for the BSc Biomedical Sciences degree programme at the university. So that's a very large undergraduate programme with a large number of students across a wide range of different degree programmes. So at Ulster, I guess we can break the cancer research down into three areas: prostate cancer, skin cancer, and childhood leukemia. So prostate cancer would be the largest area of cancer research, and certainly in terms of number of PIs that would be investigating this particular cancer. They include myself, who's interested obviously in the human microbiota and the microbiome, but we also have Dr. Declan McKenna, who's interested in microRNAs and hormone therapy. Dr. Paul Thompson, who's interested in vitamin D and retinoid pathways in prostate cancer. And my colleagues as well in the School of Pharmacy have been developing microbubble technology to deliver drugs to the prostate and also to pancreatic cancer tissue. And around that, we also have colleagues who have expertise in epigenetics, and within our nutrition centre in Niche, we also have colleagues who also feed into aspects of our prostate cancer research. Skin cancer, Paul Thompson's also working on vitamin D and retinoid analogues in the context as well as skin cancer, although this would be a much smaller programme. And then within the Centre for Stratified Medicine, Personalised Medicine, Cal Matchett's working on personalised therapies and drug repurposing in childhood leukemia. So in terms of my own research interests, I guess as an umbrella cover, I'm really looking at human microbiota and health and disease in very general terms. Most of that over the last number of years is really just focused on skin disease and cancer, primarily prostate cancer. Those two might seem slightly detached from one another, but there is, in terms of the work we've been doing on the skin and certainly in terms of the prostate, some level of overlap. And we've also been working on the microbiome, particularly the oral microbiome, in the context of brain health. And this is an area that is expanding and probably will become a larger feature of our research. And we also do a little bit of work as well in terms of degenerative disc disease and look at anaerobic infections in discs. Okay,
Andrew, sorry, just let me know when you need me to move the slides.
It's okay. I actually can move them myself if that's okay.
No, yeah, but I'm sharing, so I need to.
All right, sorry. Yeah. Sorry, I didn't realize. Yeah, I can move the slide now, please. So, in terms of the work that we've been doing in the human microbiome and cancer, we've currently, over the last number of years, been focused on the role that human Cutibacteria may play in the development and progression of prostate cancer. And more recently, there's been a few studies suggesting that one of these organisms, Cutibacteria machinus, may also have a role to play in potentially the progression of certain gastric cancers as well. So most of the work in our lab has been based around funding we've got from Prostate Cancer UK, and a large part of the funding as well has come from European and local funding that was part of a large grant startup package for the Northern Ireland Centre for Stratified Medicine, of which I'm also affiliated. And we've also published a number of other studies focused on the diagnosis and improvement in patients with cervical cancer in relation to HPV, although that's not an area that we focus too much on. It's really the prostate cancer that we've been working on over the last number of years. Next slide, please. So really just research highlights in terms of what we've found. So the key highlights from some of the work that we've been doing is we have found that this organism, Cutibacteria machinus, these Cutibacteria are anaerobes. We found a very strong association of C. acnes, Cutibacteria machinus, with prostate cancer tissue versus normal or controlled tissue samples. It's a very high level of statistical significance. And we've found that this is associated with an increased risk for prostate cancer for men who have the presence of this organism in their prostate tissue. Some of the mechanistic studies that we've done have also found evidence for certain strains or lineages of C. acnes to have a heightened capacity to drive gene dysregulation, and also to initiate some evidence that there's some initiation of cellular transformation in vitro. And these organisms are found in the prostate tissue either as stromal aggregates, possibly as biofilm, and also as an intracellular pathogen within the prostate cells. And we've also, through the work that we've been doing, been able to gather together a repository of highly characterized prostate-derived C. acnes strains. So moving forward, what we want to do is to do some metagenomic mapping and look at that in the context of clinical outcomes. And we have our own in-house sequencing platform to do this called QutusSeq. We also want to expand the mechanistic studies that we're doing and also potentially look at these organisms, particularly in the context of androgen response. Animal models of prostate infection is something we're really keen to develop and explore, because it's not something that we've looked at so far in relation to this particular topic. And then improve diagnostics, because currently the only way we can identify the presence of this organism in the prostate is by having access to prostate tissue. So we'd like to look at other ways potentially of identifying the presence of the organism in the tissue in a less invasive way. And we've been working with a number of different universities in the context of this. We're working with Aarhus University and Umeå University as well, who are also interested in this particular topic. Next slide, please. So in terms of infrastructure at Ulster, we have a number of different core facilities. So we have a bioimaging core, which has I think it's 5 million state-of-the-art instrumentation for bioimaging. We have our genomics unit, which offers next generation sequencing. It offers a range of parasequencing, a range of services such as CRISPR, DNA methylation profiling analysis. We have our human intervention studies unit, where our colleagues from Niche basically can look at nutritional intervention based on with human volunteers. And we have our biomedical and behavioral research unit, which is for preclinical and physiological measurements in rodents. And we have a new cutting edge mass spectrometry centre as well. So the work that we have done so far in relation to prostate cancer has been done in collaboration with colleagues in Belfast City Hospital in the Department of Urology and Histopathology, and also with colleagues in Department of Urology and Cytopathology at Elton McGalvin Hospital in Derry, Londonderry. And we have those links, so we're very, very keen to try and expand those clinical links if possible. So that's me. I just want to take the opportunity again to thank you for the invitation to speak this morning. Thank you.
Thanks very much, Andrew. I really enjoy that. And as in the previous session, we'll relegate the questions to a joint question session at the end. So with that, we'll proceed to Ken Nally, who's going to talk to us about interferon gamma signaling and systemic spread of gut bacteria for immunotherapy. Ken, over to you.
Thanks, Paul. Can everybody see my slides okay?
Yep.
Okay. Let's see if we can get a pointer here. Second. Okay, sorry about that. Okay, so it's still morning, so good morning, everyone. Thanks for the invitation to speak and just give you a short overview of some of the research in my group, which is relevant to immuno-oncology. My research group is focused primarily on inflammatory bowel disease, essentially looking for new therapeutic targets within cytokine networks, which may provide an explanation for the therapeutic gap in inflammatory bowel disease in terms of lack of response to anti-TNFs and other targeted therapies. So my group really is focused very much on Th1 biology. So that's the first part. First few slides will be focused on interferon gamma signaling. And the second aspect is involves my interest in the immunomodulatory effects of gut bacteria, and some new data from our group, and an area of research we're developing at the moment that I think is relevant to immuno-oncology also. Okay, so just a bit of background on the left-hand side of this slide. So what we know from the last decades of immunology research in terms of cancer biology is that a cancer cell response to interferon gamma is essential for efficacy of immune checkpoint blockade inhibitors. And this is manifest in terms of resistance mechanisms, both primary and acquired resistance mechanisms, where we see defects in interferon gamma signaling pathway as well, but also in terms of other pathways that interferon gamma regulates. And this is a selection of articles in the bottom left from genome-wide screens looking at mechanisms utilized by T cells and NK cells to kill cancer cells. And essentially the two main pathways which are essential are the interferon gamma pathway and the tumor necrosis factor pathway. Indeed, clinically as well, conserved interferon gamma signaling is associated with response to immune checkpoint blockade. And again, it's associated with resistance as well. And emerging work in this area shows that interferon gamma is not just delivered in the context of an immunological synapse between NK cells and T cells and their target tumor cells, but it also seems to form some kind of concentration gradient within the tumor microenvironment. So it would seem that there's a threshold effect in terms of cancer cells responding to interferon gamma. So what we work on is the crosstalk between interferon gamma and TNF and their effects on cancer cells, but also their effects on normal epithelium in the context of Crohn's disease and immunopathogenesis in Crohn's disease. So just to show some brief data from a recent paper where we looked at a panel of colon cancer cell lines here, and we looked at the response to interferon, TNF, and the combination of interferon and dose response. And we can see that one of the lines, LOBO, is responsive in terms of a cytotoxic effect to interferon gamma. And then we see a synergism between gamma and TNF in some of the cell lines to greater or lesser extents. So this combination, which is secreted by T cells and NK cells, is very effective at killing cancer cell lines. Not all of them, some of them are resistant. We also wanted to confirm this in organoids from Crohn's disease patients, but also healthy controls. And the interesting thing about the cell death that we observe is that we can't rescue it with canonical inhibitors of cell death pathways. So if we use a combination of inhibitors for apoptosis, necroapoptosis, ferroapoptosis, and ROS, we cannot rescue this type of cell death. So it seems to be a new type of inflammatory cell death that is instigated by this JAK1/2 STAT1 pathway. Morphologically, if we look at an electron microscopy of organoids treated with this combination, we see an atypical morphology with very heavily vacuolated cells, very enlarged mitochondria. So we're very much interested in trying to understand the complexity of signaling through the interferon pathway, because we think that understanding this basic mechanism will allow us to understand both response to interferon, resistance to interferon, and maybe pseudoprogression mechanisms underpinning pseudoprogression in the context of checkpoint blockade. So areas we're looking at are mechanisms through which this pathway decodes gamma concentration gradients, the transcriptional responses to these different gamma concentration gradients, the mechanisms and mode of this atypical, unusual type of cell death, mechanisms underpinning divergent responses to gamma. So in certain cancer cell lines, gamma will induce proliferation and not cell death or senescence. And we're trying to think about ways of replicating the effect of interferon gamma with targeted drugs that would target pathways leading to cell death downstream of the signaling cascade. A related area we're interested in is the role of interferon gamma in changing a tumor microenvironment from a cold tumor microenvironment to a hot one. So it's been known for quite a while that interferon gamma regulates these CXCR3 chemokines, which are critical for recruitment of CD8 and CD4 T cells into the tumor microenvironment, and also NK cells. A very recently published study, very nice study, single cell study and spatial biology study, comparing mismatch repair proficient colon cancer, which is resistant to checkpoint blockade, with mismatch repair deficient colon cancer, which 50% of these patients respond to checkpoint blockade and would have a hotter tumor. If we look at the graphical abstract for this paper, we see on the left-hand side that both types of colon cancer share this neutrophil myeloid-rich microenvironment at the luminal interface of the tumor. The mismatch repair deficient colon cancer has these immune hotspots or hubs consisting of both cancer cells and myeloid cells, which are responsive to interferon and have an interferon-stimulated gene expression signature, and also express these CXCR3 ligands. And these tend to be co-localized with CXCL13 T cells. So these are the types of immune hubs one would like to increase in a cold tumor. And what we've been doing the last few years in our lab, even though we were working primarily on Crohn's disease, we have this project where we're working on an approach to try and come up with a targeted therapy for colon cancer. And the basic idea is if we can come up with a rational approach to rewire the T cell and myeloid chemokine network in a colon tumor so that they would be responsive to checkpoint blockade. So the idea then is you want to go from a colon tumor with a low immunoscore, which is cold, and try and transition it into a high immunoscore hot tumor. So it's going to be more responsive to immunotherapy. The approach we've taken kind of highlights some of the technologies we use in our group and our approaches. So the first thing we did was we've done some nanostring profiling of healthy colonic biopsies, UC biopsies, Crohn's disease biopsies, and tissue from colon cancer. And we've done this in collaboration with colleagues at Queen's. And again, our data from this profiling basically matches what we see here from the single cell study in terms of sporadic colon cancer having this neutrophil-rich, myeloid-rich immune populations, quite similar to ulcerative colitis. What we've done then as well, we've screened 600 ligands to try and identify ligands that suppress production of CXCR3 chemokines. And we've identified several interesting ligands, which we've then characterized in both cancer cell lines, and we've also validated these in organoid lines from healthies, Crohn's disease patients, and CRC patients, where we've basically tried to ensure that the targets we've selected that suppress CXCR3 chemokines do it across these models. We've also done this in the MC38 tumor model, which we've used in a preclinical study. So this is a 4-year project that's just recently completed. We have the data from our Syngenic MC38 model, and we're just trying to wrap up this project at the moment. So the last slide then I have in terms of our research interests that are relevant to immune checkpoint blockade and immunotherapy is to do with the phenomenon of the association between the gut microbiome and resistance or efficacy of immune checkpoint blockade in patients. So there's 2 papers just shown here at the top, which these are landmark papers which kind of have shown or prove what's been known for 100 years. Firstly, that there's many human tumors have their own intrinsic microbiome, and it's composed of tumor type specific intracellular bacteria. Another recent paper in Science, kind of reiterating what Paul showed from his work, is that dietary fiber is beneficial in terms of response to immune checkpoint blockade, and that not all probiotics are the same. Some probiotics have a negative effect when it comes to preclinical models and response to immune checkpoint blockade. So this biology, again, is part of a wider emerging biology in terms of how actually the gut microbiome affects biology in extraintestinal tissues. And the key question we had the last couple of years in our group is how do bugs in the gut exert immunomodulatory effects in extraintestinal tissues, including tumors, and how if this affects response to immune checkpoint blockade. So recently we decided just to look basically at whether or not we could detect live bacteria in tissues from both wild type and MYD88 knockout mice. And indeed, we were able to find gut bacteria that were present inside cells in several systemic tissues. We sorted these cells using flow cytometry, and were able to isolate strains. So here we see some data from some of the strains we've isolated from systemic tissues. And basically what we see here is that these strains have an ability to persist inside dendritic cells as live bacterial cells. So in our group at the moment, we've identified mechanisms which gut bacteria use to disseminate to systemic tissues. These bacteria exert atypical immunomodulatory effects on immune cells. They seem to be able to enhance migratory effects on immune cells. And we're currently establishing cell-based screens for bacteria that induce migratory behavior in immune cells and immunomodulatory behavior in immune cells. Our goal for all of this work and this future work is to test strains for efficacy with immune checkpoint blockade in Syngenic murine tumor models, and also to test these strains for efficacy in murine models of infection and inflammation. So just to close then, at the APC, we have several technologies available which support our human studies, both in the context of host microbe interaction, inflammatory bowel disease, and tumor immunology. So we have a flow cytometry platform with analyzer sorters. We have a 10x platform as well. And as Subrata mentioned, thanks to his success in terms of his SFI professorship, we will have an imaging mass cytometry platform soon. We have a very good human studies platform that helps us to collaborate with our clinical colleagues. We also have hypoxia and anaerobic chambers, and we've established human gut organoid technology at the moment primarily for studies on inflammatory bowel disease, but we're also starting to use this for colon cancer research. Okay, so thanks for your attention, and take some questions.
Thanks a lot, Ken. That was great. We will drive on. We'll take the last talk, which will be delivered by Professor Joachim Pren, who's well known to most people in the cancer ecology in Ireland. So over to you, Joachim. Your sound is problematic. Try a sound test. Now, if you're on a Bluetooth mic, it's not working. Can you get a wired mic or go direct to the laptop mic? He's leaving. Okay, hopefully he'll be right back. Subrata, you look like you're about to say something profound to fill the dead air.
I was just wondering whether he's back.
He's back.
Hi, can you hear me now?
Oh,
that's
yeah.
perfect. Yeah, thanks.
Yes, that's what my teams, I have to start it twice, and I never
Yeah.
understood why. So I just, first of all, like to thank everybody for organizing this meeting, particularly my program officer, Isabella, who was really behind organizing it, and Jarushka and Paul for putting all these nice speakers together. So big thanks to all of you, of course, to Rosemary and Liam as well. I just want to brief, hopefully it works now. Oops, that's the wrong. Okay, can you see my presentation now?
Perfect, thanks. Yeah.
Okay, excellent. So I'm based at the Royal College of Surgeons. I'm the director of the Center for Systems Medicine here at RCSI, and we have an interest, of course, in systems approaches, particularly in the field of cancer and colorectal cancer. And I, like Paul myself, would have never thought that I would present anything on microbiomes ever in my lifetime as an academic, but here we are. And the reason for this is that an older paper of mine that I published with David Hughes in 2014 actually really took off, and is one of my most cited papers that I have ever authored. And this is a paper we published in a quite obscure journal for me at the time, the European Journal of Clinical Microbiology and Infectious Diseases. And this is work we did in collaboration with centers in Germany and the Czech Republic, where we for the first time really showed that Fusobacterium nucleatum is associated with disease progression in colorectal cancer, and also with disease outcome. Can you actually see my mouse? Can you see my mouse when I move my mouse?
Yeah, we can see the cursor, yeah, on the left panel, yeah.
Yeah, okay, excellent. Okay, so these are just shows you what this really early work in 2014, what we have shown here. These are normal and matched tumor samples, and this here we quantified Fusobacterium nucleatum with PCR assays. We could actually here detect an increase in Fusobacterium levels in tumor tissues in those patients from these 3 independent European cohorts. We also looked at the cancer progression, where we found that really pre-stages of cancer or early dysplasias, adenomas, do not show this increase in Fusobacterium load in the disease tissues. So this is really an evidence for us that this was really an indicator of potential disease progression. And we did with quite a low number of patients, where we also had some follow-up data. We did, a very preliminary Kaplan-Meier, and saw also that the prognosis of patients with high Fusobacterium load was much worse than those patients with zero or with low Fusobacterium levels. Of course, as probably Paul and everybody else in the field knows, there has been quite some discussion as well as to whether Fusobacterium was just a bystander effect, whether it's just a sign, of inflammation, and or whether it's actually also contributing to pathogenesis. And there has been, of course, some key papers involved produced in this. So we basically, after all this sudden interest back into our Fusobacterium story, we just basically looked into data from the TCGA, but also with a nice collaboration with the Queen's University Belfast, with Daniel Longley, and the taxonomy cohort that some of you might also be quite familiar with, as part of our NIH US tripartite grant. And what we showed here, we just published this last year, and I don't think it's out yet in press, is that we do see here that, in fact, the association of Fusobacterium nucleatum or Fusobacterialis more generally with the disease progression seems to be linked solely to patients that have what we call a mesenchymal or CMS4 status. So this is some data of that paper here, where you can see that, in fact, you can subtype colon cancers into 4 different subtypes called CMS 1, 2, 3, and 4, consensus molecular subtypes 1, 2, 3, and 4. And it's known that CMS 1, which is immune-rich, has high levels of Fusobacterium. That is a known fact, and that we can also confirm both in the taxonomy as well as the TCGA cohort. This was PCR based, this was a PARSEQ based analysis, and compared to other subtypes, they're quite low, in particular also to the CMS4 subtype. But the interesting part is that really in the CMS4, it really mattered whether or not the Fusobacterium load had any impact on disease progression. In fact, it was the mesenchymal patients where Fusobacterium had a negative impact. As part of this work, we also, of course, then do what we call a multi-omic characterization of association of Fusobacterialis infection with tumor signatures, which could be, sequence genomic alterations in the tumor, RNA sequencing data, or RPBA proteomics data. But the really interesting thing that we wanted to explore really is why it is that just these mesenchymal subtypes seem to have bad prognosis if there is a Fusobacterialis prevalence, a high Fusobacterialis prevalence. So we did a systems approach, a logistic regression analysis, really to model this three-way association between Fusobacterium load, molecular subtype, and all this host contexture, i.e. all the omics data. And what we actually found identified that it seems to be in the CMS4 patient that there really are really strong aberrations in a couple of cytokines and chemokines that actually make certain macrophages and other immune cells go, really get them on the move. And these are particularly Notch, CSF1, CSF3, and the interleukin 6 and interleukin 8. And we propose these as novel candidate targets. And of course, going forward, we would be interested maybe also working with an animal group, such as or with other groups that have presented the work here today to really go into also functional studies going forward. So where are we else going forward? So we also have an interest in multiplexing, hyperplexing of tissue samples. And we now have also received a grant, an award from Science Foundation Ireland under the Research Infrastructure Program and the NASPRO, the National Spatial Tissue Profiling Platform, coordinated by Liam. And we will are currently in the process of acquiring a hyperplexing platform, particularly the CellDive platform that we have worked with under the NIH tripartite grant for quite some time. And just to show you some of the initial staining, what we can do, for example, we can basically take this word taxonomy tissue samples from this clinical cohort where we had PCR data on Fuso high versus Fuso low patients. You can see here that the Fuso high patients also stain with a specific antibody to Fusobacterium in the tumor tissues here. This is a Fuso low by PCR, which has no staining. And we then basically can also see an association of different, immune cells, beta-catenin, differences in staining, and so forth. So of the aims, what we really want to do going forward is to identify tumor and other cell types that are infected with Fusobacterium nucleatum, for example. Then using the hyperplexing platform, identify the cell state of infected tumor cells or other tumor microenvironment cells. Which basically means that we will look at antibodies that recognize, let's say, an inflammatory state, in poor apoptotic state, cell survival state, an EMT state, and so forth. Because we can use up to 40 or 50 different antibodies on the same tissue section. So this is what we refer to as hyperplexing. And we have, through our NIH grant now, also developed the image analysis pipelines for detection or for analysis of these huge data sets. And of course, we also, with this, we can also detect spatial associations, i.e., local effects of Fusobacterium infections that you would not be able to see as rapidly, for example, by using single-cell RNA sequencing or other approaches. The other thing we do, and this goes back a bit more to our systems biology research, is that we also have now identified actually microbiome subtypes in colorectal cancer based on both bacteriome and microbiome data. We can identify 4 distinct subtypes that are quite clearly separated, so quite nicely separated microbiome subtypes. And these subtypes are also preserved across different cancers, also across lung cancer and other cancers. And more important, we find that these subtypes also have a strong effect on prognosis of patients, not just in colon cancer, but also across really all other cancers, really like a pan-cancer subtype that we detect. And this paper is also currently in publication that also basically aligns with some of our systems biology work in our center here. And likely we're also going to look at association of signaling pathways in these 4 subtypes. So this is just my brief introduction on our microbiome research that also links, of course, with inflammation research. And if you have any interest contacting me, interest in collaboration as well, my email is there shown. And yeah, with this, I really want to conclude and thank again Isabella for organizing this meeting. Thank you.
Thank you, Jochen, for a fascinating presentation. I really like the stuff towards the end there. In fact, I like a lot of what I heard after lunch, or just before lunch, rather. I think that there were some resonating themes between the 2 sessions in terms of we've got some excellent basic science and challenges associated with translational and connections between basic science and clinical science. And in terms of immunotherapy, Jarushka has mentioned the desire to establish a national protocol. I think that would be important and relevant because we don't have an Irish immunotherapy cohort where we can identify nation-specific variations. Jochen has very nicely introduced a pan-cancer microbiome, which excitingly translates across different cancer types and is presumably a reflection of different immune statuses in patients with different kinds of cancers, and it includes the microbiome, and that's great because the microbiome does not seem to be relevant for other diseases we've looked at. I don't see specific questions in the chat, but I see Liam has his hand up. So Liam, what would you like to contribute?
I think Subrata could have been there before me, but
oh, sorry.
Go ahead. Go ahead.
Yeah.
Liam, and I'll come back later. Yep, that's fine.
One of the things we were kind of interested in, I'm just wondering if there's been much work in that area, is looking at the spatial relationship between the bacterial species in tumor tissues and immune cells. So I'm just wondering, obviously, because of the different platforms which are now available, under the National Spatial Profiling Platform and then the system that Subrata mentioned, the Hyperion system, is there an opportunity to do something where we connect the dots there in the 2 areas where, now maybe it's too complex, where, using an in situ approach where you can actually look at different bacterial species in tumor tissues and then look at their relationship with different immune cell subsets. I'm just wondering, is that of interest, or is there any kind of relevance?
I think it can be of great interest, and in fact, it is of great interest. And I think understanding the relationships between these are key to understanding the pathogenesis, but also many other things we spoke about. Remember that there was this tremendous excitement when Nature published their cover with the 3 microbes biofilming across a colon cancer, and it was a spectacular picture that attracted many researchers. So I fully agree with you, and I think we should also actually make it an integrated platform, with your platform, and for the AICRI to access it for teams. What I was going to say, I think, which actually ties in very well with it, is I do think that regarding immunotherapy, understanding either the breaks or the escapes that lead to resistance, where the microbiome is involved and the immune cells are involved, and many of these immune cells, even for cancers outside the gut, are in the gut, as is the microbiome. And I personally think that there is a lot of mileage in AICRI investing in very focused research in understanding this, the brake mechanisms and the brake circuitry and the escape circuitry that relate to immunotherapy non-responders. And for this, I think we need a different approach. I do not think that the current way of collecting biomaterials is sufficient. In order to understand resistance or escape or breaks, and to understand the evolution of the circuitry, it is a very dynamic process. It's not a static circuitry that stays there and just drives it. It actually evolves in response to therapies, immune therapies of all kinds, cancer, chronic inflammation. And we need reagent standard observational studies that are highly protocolized to get biomaterials. Just a baseline sampling or endpoint sampling. I think we should sample less people suffering from cancer, but we should do deep reagent standard sampling in this fewer patients. This is the only way to succeed.
Thanks, Subrata. I see Liz Ryan has her hand up. Liz, would you like to ask a question?
Well, actually, I just want to underline exactly what Subrata has said. The idea that you can take one biopsy and measure PD-1 as a predictor of response is really not based on any understanding of immunology, because the microenvironment is so dynamic. It changes all the time. And as soon as you give a treatment, you're changing the immune system. We see it with our inflammatory bowel disease patients. If you sample them pre-TNF treatment and after TNF treatment, you change their immune phenotype, you change their ability to respond to immune stimulations. So the idea that one sample and one biopsy can answer these questions is very naive in a way. It's because this is so dynamic and the cells traffic and things and receptors go up and down and the microenvironment changes. So I just really want to agree very strongly with that.
Yes, maybe I can say something there. So in terms of, I would be very interested to look at not only Fusobacterium nucleatum, but potentially also other species we and others have identified to be associated with cancer progression in colon and other cancers. I think it's just one. There's a couple of them popping up now. Established cross-board maybe platforms where we can use like our cell dive technology, hyperplexing approaches, and look into this in terms of spatial analysis as well. And that's exactly, I just submitted an NIH grant, which didn't get funded again, by the way. I have to resubmit it again with my NIH team. So unfortunately, just exactly on that topic, looking at the spatial association and our idea that we macrophages, we get on the move, get kicked to go really by Fusobacterium in particular. But I think the issue we have is really the samples and getting pre-treatment samples, or even best pre- and post-treatment samples, and the size of those samples that will be very difficult to obtain. I talked with Jarushka about the lung cancer, where we also see this Fusobacterium nucleatum signature very strongly as well. Getting pre- and post-treatment samples, or any samples at all, is not straightforward. And I think one way how we can maybe promote this really, because we really need FFPE sections anymore, so we don't need really fresh frozen for many of our approaches anymore. It's all about FFPE going forward. We'll try to see on a platform, a national platform, and I would be quite open to, if it's pancreatic cancer or esophageal cancer or lung cancer or colorectal cancer, just once we have these platforms, these techniques established, we can address these key questions, quite in a quite elegant way. And I think that could be a strength that we have in Ireland, is combining the immunology, immuno-oncology, and the microbiome research and the excellent in our centers that we have across the island going forward. I think that's something I want to establish, and maybe want to use the AICRI funding we have also for somebody to establish exactly that.
Thanks,
I can come in there and, and sort of say, yes, we've written the protocol. I'm happy to adapt it. There's a couple of people here who've been, involved in that. I think, as Subrata was saying, and Donal previously, focusing on 2 or 3 very specific questions, and probably on, say, 2 specific tumor types, is probably going to minimize the heterogeneity and help us to focus. I think obviously lung cancer is a great one. There's not a lot written on it. There's a lot of dynamic lung cancer researchers open to hearing about other tumor types. I think the other area that clearly a number of us are interested in is immune toxicity. Subrata, Elizabeth, myself, we have a deep experience of this, and we could leverage the experience of our colleagues who are not oncologists. And that is quite a niche question, that I don't think many groups in the world are actually studying in a very collaborative way necessarily, compared to maybe acquired resistance, where I think there are many, many labs exploring acquired resistance or even primary resistance to IO. So I guess a little bit of strategy regarding which question we want to go after, because as a small country, I think we should really focus.
Agreed. Andrew McDowell, in the context of Cutibacterium acnes and prostate cancer, which is really intriguing, if I want to action that and have a surveillance program, what samples do I need, and how does the bacterium get into the prostate tissue?
Well, answer the second question first. I don't think we're absolutely sure exactly how it gets in, but it's not just found. I mean, it's a predominant organism on the skin, but it's not just on the skin. It's also in the oral cavity, it's in the large intestine, and it's also present in the genitourinary tract. So I guess our current assumption is that it's getting in via the colonizing. It's already in the genitourinary tract, and it's making its way up into the prostate. That's what we think, and it seems to be the most logical route into the tissue. In terms of, sorry, your first question, I think just in terms of what we need to do with patients. So there have been a number of studies showing that, certainly our work has showed that there's the presence of this organism in prostate tissue. It has a very, very high specificity for prostate cancer. So you generally find when the organism is found, it's generally found when there's a tumor present. The sensitivity is lower, much lower as you would expect, because not everybody who has prostate cancer will have this organism. So there has been some suggestion that, when we do a biopsy, if we're also looking for the presence of the organism along with the normal, that might have, diagnostic value. Either if we find that there's not a tumor present, that maybe we've either missed it, or as a follow-up with that particular patient, that we need to keep an eye on them and maybe see them maybe more frequently, just to monitor their conditions. I think it was a Japanese study which showed that, and I think it fits in with the sort of the data that we currently have. I guess where we were coming from was, if this organism is causing chronic, long-standing asymptomatic infection, which we believe it may be doing, what we want to do is try and identify it and maybe hit it before cancer develops. The problem there is, how do we identify it in a man who doesn't have prostate cancer? The only way we can really identify it at the moment is through getting hands-on tissue. So we're stuck in this kind of dilemma. So that's why we were kind of interested in looking at maybe what less or non-invasive or other ways of maybe trying to identify or matching up, certain biological parameters with, biomarkers, maybe in urine or seminal fluid or something like that we could access. So that's kind of where we were coming from. There's a vaccine being developed for this organism in relation to acne and also in relation to prosthetic joint infections. It's a therapeutic vaccine, so you could kind of see that maybe that could be an approach. Eradication therapy could be approach. Again, the problem with the eradication therapy is, I think sometimes the prostate can be difficult to get antibiotics into the prostate, and the other issue is, even if you eradicate it, who's to say that it couldn't down the line reestablish itself again, because it's part of the host microbiota. So there's these kind of things that we've thought about, not exactly got the answer to, but I think there's, in terms of the translation of the research, there's a lot of potential there. Certainly based on the data that we haven't published yet, but we think that this is a real, the organism is present, it's not a contaminant. The data that we've got, certainly in vitro at the moment, shows it clearly has, certain strains have the potential to drive, certainly in vitro, cancerous changes or changes that have the potential to turn the cell into cancerous. So, there's a lot of data still to develop in this organism, but it's I think from a translational point of view, it's very very exciting area. Just to finish, there is other organisms present in the prostate. We know that, but transient colonizers coming and going. It's that's the nature of the prostate. But this organism is positively associated with chronic and acute changes. A lot of the other organisms aren't, so they're probably not clinically relevant. So that's kind of where we're coming from. But it's not, I'm sure, the only show in town, but we think it is a significant player, and we find it in about 35% of cancerous prostates versus less than, I think, 9% of normal prostate tissue.
I think it's a great story, Andrew, and in the context of profiling AICRI as an all-Ireland discovery platform, which ultimately is to benefit patients, it sits really nicely in the spectrum of basic and fundamental research through to translational and discovery research, which could lead to screens and interventions to reduce the incidence of a cancer with very poor prognosis typically. So I really enjoy that part. Thanks, Andrew. Ken, did you want to come back in? I'm going to wrap it up in the next couple of minutes so we can get some sort of lunch. Ken?
Yeah, no, I mean, I guess, yeah, relating to that discussion, I'm just curious about this idea that bugs from different tissue sites can circulate, you know, that there's an active mechanism the bug is utilizing to get from a distant site to a tumor. So I guess, Andrew, have you looked in blood from patients to see if you can detect the organism in blood?
Part of the problem is when you take a blood sample, you put the needle through the skin, so you generally will get evidence of the QD bacteria, the propionic bacteria in the blood, and obviously propionic bacteria in blood products is an issue. It's not something that we have looked at, but we have been interested in the idea of the concept of the blood microbiome. Whether it's fact or fiction, I don't know, but that blood microbiome, I think is it's a bit like a motorway that can transport things probably from, particularly in the gut and the oral microbiota as well, and to distal sites. But we haven't really looked at blood. We haven't looked at that in any real context. Our assumption has primarily been that this bacterium is probably present in the genitourinary tract of some men, and it can, you know, ascend up into the prostate. There may be other mechanisms that we're not aware of, but that's currently where we are at the moment.
Okay, right. Thanks, Andrew. And I guess this is for Paul. I should know this, but Paul, like in that idea of a blood microbiome, have people looked at that in detail in terms of cell-associated host cell-associated microbiome and microbiome in the blood itself, in the serum or plasma?
They have, and there are companies actively working on it. And I have a friend in Glasgow, Paul Shields, who's looked at it quite a lot. It seems that it suffers from the transient nature of transient bacteremias, which happen with local insults to the colon or other tissues where you can get access, or when like transient bacteremias from brushing your teeth. So it seems it's quite variable, and from what I've seen, it's not a reliable biomarker for much. In terms of cell association, no, apart from your kind of work, there's not a whole lot.
Yeah, I mean, our work, which is kind of unpublished, but we have good evidence now for cell-mediated transfer, so host cell-mediated transfer of a bacteria. So yeah. I'm just curious, you know, if there was some type of a focus project, it would be interesting to test that idea that some organisms can actually manipulate host cells for trafficking. Yeah. I think it might be interesting to look at blood cell associated microbiome, both in terms of, I guess, metagenomics, 16S, or trying to culture bugs. Yep. Has anybody ever tried to culture bugs from PBMCs, anything like that?
Yeah, they have, sure. That's doable, technically doable. We can add that to the long list of things we'd love to do under the heading of AICRI. I think we better wrap it up, and we're behind schedule, but not terribly so. So I propose that we resume at 12:50, which gives us a 15-20-minute break for lunch. So we'll resume 5 minutes late at 12:50 for the next session on funding and implementation. Okay, so thanks everybody, and the same link will apply for lunch. Thanks, Subrata, for wonderful co-chairing, and to all the presenters. Enjoy your lunch. Bon appetit.
Okay, thank you.
Thank you.